Welcome to our second podcast on the nervous system. What we'll be doing in this second podcast is we'll be looking at neurons and neuroglial cells. So we'll classify the neurons based on their cellular structure, principally the number of dendrites, axons, but then we'll also describe each type of neuroglial cell, especially with their histological appearance as well as function. And then lastly, we'll define the process of myelination and look at all of the fine structures that you can identify when we look at a myelin sheath. In the first podcast, we talked about the soma, the axons, and the dendrites of your generic neuron. Well, now we're going to put this together and to look at all the different shapes that neurons can accept and have in the human body. One of the common types of neurons are the bipolar neurons, and one of the places that we're going to see these bipolar neurons is in your olfactory epithelium. So as we look at our bipolar neuron, we can see that we have a central cell body. We have one somewhat thick dendrite, much thicker than we're used to seeing, and remember this is bringing information to the cell body, and then we have a single axon leaving the cell body. Here we have our olfactory epithelium, and can you classify this for me? It's olfactory, so we're in the respiratory system, and even though we can't see it so well, if we look at this, that is definitely pseudostratified, and we have cilia here at the surface. This particular cell in this sea of nuclei, this is going to be one of our bipolar neurons, and we can see the dendrite, and then we lose the axon as the axon extends down. This is going to be just one of our glands, ducts of our glands that we have coming up, and we'll talk about that when we get to the olfactory system. But in fact, when you look at olfactory epithelium, these bipolar neurons and their axons that extend down, and they're going to extend down, and they're going to be going toward, a synaptic point in which we are going to bundle those cells together. Each one of these axons are going to comprise what is collectively known as the olfactory nerve. Now, the next type of neuron is a unipolar neuron, and some people call it pseudo-unipolar neuron because, in fact, it doesn't just have one. It does have an axon, and it has a dendrite. But its shape is going to appear to have one long stalk off of which you have a perpendicular process going to the cell body. Now, these are going to be typical of our sensory neurons, and we're going to see these cell bodies present in our dorsal root ganglia and in some of our cranial nerve ganglia where we'll find these. So here we have our pseudo-unipolar. You can see it appears to have one process and one process going to the soma. However, this is the dendrite, and once we make this turn, this is actually two-way traffic. We'll see the dendrite goes here, and then the portion of the axon comes out this way. This is going to be the cell body that's located in the ganglia. And remember, the term ganglia refers to a collection of nerve cell bodies in the peripheral nervous system, whereas a nucleus is a collection of nerve cell bodies in the central nervous system. In our histological view, we can see our typical cell body of a neuron. Nice round cell body. We've got our nucleus and the prominent nucleolus in the very center. Now, the most common type of neuron is going to be the multipolar neuron. These are essentially the alpha motor neurons and the gamma motor neurons that we see. Typically the cell body with a number of dendrites and a long single axon extending away. Now, that's the textbook drawing of what a multipolar neuron is, but sometimes it's a bit oversimplified because, as you can see, you can have a number of different processes, and all of these are going to be cells that have been identified. This is your stellate neuron, stellate meaning star-shaped in this case, dendrites, there's the axon. Here we have a pyramidal neuron, and we'll be looking at these as we get into the cerebellum and the cerebrum, and especially looking at that rather triangular shaped soma. More than anything else, we're going to be identifying the soma of these neurons. But as you can see, you have many different sizes and shapes of these neurons, multiple dendrites, one axon, possibly with collateral branches, but these are all our multipolar type of neurons. So once again, here we have a histological stain. This is going to be a multipolar neuron. We see the cell body of this neuron. We can see the nucleus, the nucleolus. We have Nissl material in here. And then we have these processes extending away. And I'm hard-pressed to tell you where the axon would be. We see a lighter staining region here, but there seems to be two processes extending away, so I'm hesitant to call this an axon. The axon possibly is out of the plane of section of this particular neuron. Now, in the peripheral nervous system, our nerves bundle themselves together into peripheral nerves. And when we look at the spinal cord and the nerves adjacent to the spinal cord, we have sensory fibers going to the spinal cord and motor fibers running away. They bundle together into a spinal nerve. And when we look at the organization of these are going to be fairly similar to how muscle cells are bundled in skeletal muscle. And we're going to have connective tissue wrapping that's going to essentially bundle them together in the same way with very similar terms. So as we look at a neuron, the connective tissue that is going to wrap around the neuron, which is essentially going to be with the basal lamina of the Schwann cell, this loose connective tissue is going to be the endoneurion. Then, as we bundle multiple axons together, we're going to have that connective tissue around it. This is going to be dense, irregular. We're going to bundle that together into the perineurium, creating fascicles. Again, this sounds very similar to what we did in muscle. And then lastly, the entire nerve is going to be wrapped in this dense, irregular connective tissue with the epineurium. And as we look at the illustration, if we didn't know that this was axons and dendrites, you might just suspect that we're looking at a muscle, where we have individual muscle cells with the endoneurium. Then we have our fascicles wrapped with the perineurium, and on the very outer portion, we have the epineurium. Here's our histological stain of a fascicle. Again, around each one of these axons would be the endo. This is going to be the perineurium. And again, I'm avoiding the edge out here. They still show this as perineurium. But out here in the very edge of our nerve, this would be the epineurium. I know that's hard to see. You just get a little slice of it here. But since you've got other fascicles here and here and here, I don't see them up here. I'm betting this is going to be the epineurium. On an actual practical or quiz, you would see more than just this. Now, neurons make up only 80% of the mass of our nervous system. And so the other 80% are going to be the supportive cells of the body. And so these are collectively called neuroglial cells. And neuroglia literally means nerve glue. So we're going to walk through these, be able to identify them, and know their functions. And the first we're going to start with are the astrocytes, not the least of which reason we're starting with them. They're the largest of the neuroglial cells. These are going to be extremely important in the nervous system, particularly the central nervous system, because this is where we are going to create the blood-brain barrier. So we really create an immune-isolated environment so that we don't get pathogens, infectious agents into the central nervous system. Now, we can distinguish our neuroglial cells from one another, and specifically with the astrocytes, when we look at intermediate filaments. We're used to the desmins, the vimentins, the keratins. Well, here in astrocytes, we have a new one that is called GFAP, glial fibrillar acidic protein, and that will be a marker for any of the astrocytes. So if a patient has a tumor and you stain for GFAP, then you realize you have an astrocytoma. So again, the principal function that we have for our astrocytes, we are going to be looking at maintaining that blood-brain barrier in the central nervous system. We're also going to help maintain the microenvironment around neurons. And we're going to be able to also release stores of glucose to the neurons in certain instances. So here we have some astrocytes, astro meaning star-shaped, and I think you can see why we use that terminology with these astrocytes. Again, in a light microscopic field, you're going to be hard-pressed to really see these and distinguish these from one another. So what we're talking about right now is more functional than we're going to be doing for visual identification for the nervous system. Now, they can appear in two forms. You can either have a protoplasmic, which are going to have a number of processes. They're almost going to look hairy. There's going to be so many. Or a fibrous one, which are going to have fewer processes, but they're going to be much longer, more prominent, even though they're fewer. So really with the protoplasmic, this star-shaped appearance, these are the ones that are going to make contact with blood vessels, especially capillaries, and they're going to have these pedicels, these vascular feet that are going to contact and wrap around the capillaries, and they're also going to contact neurons. So that material that needs to be passed between the neuron and the circulatory system must pass and be screened by an astrocyte before it gets into the neuron. That is one of the ways we create the blood-brain barrier. So here we can see the protoplasmic type on the left versus the fibrous type. And again, you can see how they are contacting the blood vessel. What you don't see in this illustration is just simply how they pass. The other processes are connected to a neuron. The next type of microglial cell we have, these are going to be essentially macrophages. And so when you look at their lineage, where they're derived from, they're derived from the mononuclear phagocytic system. This is where we get our monocytes, our macrophages, and so these are, for all intents and purposes, macrophages. They're going to phagocytose material, antigen-presenting cells, secrete cytokines. So these are the macrophages of the nervous system. So in this case, they look somewhat fibrous, but when you look at an actual histological diagram, we're going to be able to see these microglial cells. And again, as we're going to see in many other tissues, you're going to be able to see vesiculated cytoplasm because they are gobbling up materials, cellular debris, any degenerating cells. They're really going to begin to pick that up. But as far as recognizing and distinguishing oligodendrocytes from an astrocyte in this particular stain, I know you look at that and go, how can I tell them apart? Well, you can't. We're not going to be doing that in this particular type of stain in this slide. Third type of neuroglial cell, the ependymal cells. These cells are going to function in creating the cerebrospinal fluid. Now, these cells are prominent in the central nervous system, especially lining the ventricles of the brain, the central canal, and they're produced by a structure called the choroid plexus. They're going to be ciliated, and this helps create a current with which the cerebrospinal fluid moves. This also assists in the protective barrier, in the sense that pathogens must gain access through, so it's like swimming across a river to get to the central nervous system. But also the cerebrospinal fluid is going to be somewhat of a hydraulic cushion and a protection and a shock absorber for the sensitive underlying nervous system tissue. What we're looking at here is we're looking at neuronal tissue in the central nervous system, and we actually have ependymal cells lining one of our cavities. Again, this is fairly high mag view, but these ependymal cells are going to be somewhat simple cuboidal, possibly taller to simple columnar in nature. But in this particular instance, we are looking at simple cuboidal epithelium, again, with our neuronal tissue underneath. But you would need a much higher magnification view to know you're in nervous system tissue to be able to recognize this simple cuboidal look, to recognize that as ependymal cells. Now, oligodendrocytes, we're pretty familiar with these from histology and looking at action potentials and how the myelinated axons really speed up the transmission of that signal. I think we're also pretty familiar with oligodendrocytes occur in the central nervous system only. So when we look in the central nervous system, a single oligodendrocyte is going to be capable of myelinating a number of different axons. Oligodendrocytes, there's no connective tissue, there's no basal lamina with these cells. They simply make contact with the axon, extend several processes, and begin to wrap their plasma membrane around and around and around the axons, creating the myelin sheath. And again, these neuroglial cells, we are not going to be identifying them in histological slides for lab. We're understanding their function here in lecture. And again, expanding on what we've done in our physiology course. What we're looking at in the very center, the largest cell that you're really going to see in the central nervous system as far as nucleus goes, this is going to be the oligodendrocyte. I think it's also going to rival what we might see with the astrocytes, but we can't really see the processes in this particular stain. But here we can see the processes of this oligodendrocyte extending these processes around. What we don't detect, and we're not really seeing here, is the connection of these processes to a particular axon and then the myelin sheath that's being wrapped around. Schwann cells are the peripheral nervous system analog of the oligodendrocytes. These myelinate the axons in the peripheral nervous system. And recall, one Schwann cell myelinates one portion of an axon only. It's not going to have these processes that extend to adjacent axons. Now, another interesting difference between Schwann cells and oligodendrocytes, Schwann cells do have a basal lamina. You're going to have a basal lamina around the axon and around the Schwann cell. So in terms of peripheral nerve injury, even though the Schwann cells and the neuron might degenerate, you're still going to have this basal lamina tube that's going to remain, and that's going to help guide a regenerating axon, and we'll talk about that in our third podcast on the nervous system. But that's the big difference. Oligodendrocytes don't possess this basal lamina, but Schwann cells do. So here we have an example of a Schwann cell. We can see the Schwann cell nucleus. And then we have this interesting looking feature at the light level. This is going to be the axon in the center. It's fairly homogeneously stained. But now when we look at this region, it looks like we have spokes on a bicycle wheel. And that's actually the phospholipid of this myelin sheath being unable to pick up all the stain as well as the protein in the cytoplasm of the axon. So it's going to give this sort of spoke-like appearance where we see the myelin sheath. And you can see that's fairly thick. Now, a lot of what we're going to see and know about myelination in Schwann cells, we see at the electron microscopic level. So here we have the nucleus of a Schwann cell, the cytoplasm, and here we have this really dark, intensely stained myelin sheath. Now, as the myelin sheath is laid down, this essentially the plasma membrane is going to wrap around and around and around. And so we're going to have regions of this myelin sheath where we have opposing cytoplasmic faces of the phospholipid bilayer pushed against each other. We've squeezed the cytoplasm out. So these opposing faces, the cytoplasmic faces, will be together. And then we will have alternating layers where we have opposing extracellular portions. And we're going to give these terms, we'll have to look at it in a little higher magnification as we do, but we give these different lines names as we go through and look at them. So when we look at the myelinated portion of an axon, we can look at the internodal segments. Now understand the node of Ranvier is where we don't have myelin. So the internodal segments, that's the part of the axon that's covered by the myelin. All right, and we can call that the myelin sheath, but each one is called an internodal segment of myelin if you want to include that. Now, as we look at these cytoplasmic versus extracellular faces being pushed tightly together, the cytoplasmic surfaces together are going to create what's called the major dense line. It's going to be darker and more continuous in appearance, and that's going to alternate with the intraperiod line, which are the extracellular faces of the plasma membrane pushed together. And we'll see that in the next image. But we also have these features, the mesaxon. The internal mesaxon is going to be that part of the cytoplasm, a little bit of the cytoplasm of the Schwann cell in contact with the axon. And the external mesaxon is going to be that portion of the Schwann cell body that is on the outside, so that portion of the myelin sheath that's directly in contact with the Schwann cell body. So in this illustration, we can see, and they're highlighted here in red, we've got the axon on the inside, Here we have the cytoplasm of the Schwann cell. We've got our collagen fibers in cross-section. And you can see these alternating lines. The dark ones that you see are pretty easy to see, and those are the major dense lines. But do you see interspersed between those these really, really faint hair-like lines, and they almost look discontinuous? Those are going to be the intra-period lines. Now, we don't see the internal mesaxon in this one, but here we see where we finished that last layer, and now we've got the Schwann cell plasma membrane coming up and going around this way. This is where they join together. This is the external mesaxon, and we will find sort of the starting point where we might have a little bit of that squeezed in here. That would be the internal mesaxon. It's just not in this plane of section. So myelination, whether we're talking about peripheral or central nervous system, the mechanisms are largely the same, even though the cells are different. And the plasma membranes of each are going to wrap approximately 50 or so times, creating a very watertight barrier in which ions are not going to be exchanged. We're only going to see that at the nodes of Ranvier, and so we're going to see that saltatory conduction as our action potential velocity increases from 1 meter per second to over 100 meters per second. Now, myelination is not complete at the time you're born. It's not at all. So motor neurons almost completely myelinated at birth. Sensory neurons take several months after birth. And portions of your central nervous system, the tracts, the commissural axons that are going across from right to left or left to right, either in the spinal cord or in the brain, those may take several years after birth before myelination is complete. So timing is everything. Motor, then sensory, and then we finish off our central nervous system as it continues to develop. Once again, we can see the diagram here of myelination, and maybe you get a little better idea looking at how we have the cytoplasmic faces coming together, and then we're going to have the extracellular faces coming together. And that's going to create the major dense line and the intraperiod line. They're showing you the mesaxon at this point. And in here, this would be the internal mesaxon. If we could see a little bit of that space, they don't have that labeled. So once again, myelination is everything. It speeds up the transmission rate, that saltatory conduction rate, over 100 meters per second. And just recall, you can live without myelination, but the discoordination that you have, the dyskinesis, is pretty bad. Your quality of life is really low. It's going to shorten your life expectancy. And multiple sclerosis is an example of an autoimmune disorder where you attack the myelination, and you have probably seen and know people with multiple sclerosis. That just gives you an example of what happens when you don't stop the signal transmission. You simply decrease the rate from 100 meters per second to in the neighborhood of 1 meter per second.